Okay. Now your tertiary structure, this involves the R groups and their properties. So, as always, it's going to get more complicated. So this is when we're going to start looking at the amino acids and all their properties here. Now before we do that, I just want to fill in some of their properties and tell you what they mean. So they mean more than what I'm going to tell you, as in there's more depth to it than that, but for the purposes of what we're looking at for today in our practical, we're just simply going to go with the simplest explanations. So we have non-polar, which as far as this goes is hydrophobic. We have polar, which is hydrophilic. And I'll go into that in a bit more detail. So we've got basic, which is positive charged. Oh no, sorry, negative charge, not positive charge. What am I doing? Negative charged. We've got acid, which is positive charged. We've got a start codon, which is always methionine. And if you're a prokaryote, because you like to be slightly awkward, so you've got F-met, which is simply methionine plus a formal group. So methionine always starts, and even if you're a prokaryote, methionine still starts, but it just has a formal group attached to it, okay? And a stop codon doesn't code for an amino acid, but it does tell it is time to stop. It equals stop. So you've already got your stop ones here. So they don't get translated into an amino acid also. You won't see them at the end of your translation, but so you won't see them as an amino acid. But they are there. They are there telling them it's time to stop and dissociate. Okie dokie. So here you've got your methionine, and let's assign some characteristics to it. So methionine is non-polar, so let's just write non-polar. And then we have F, which is phenylalanine, and that is also non-polar. And then we have G, which is glycine, and that one is also non-polar. And they're all non-polar until we hit histidine. So non-polar and non-polar. Now histidine is polar. So is C, which is cysteine. That's non-polar, non-polar. And then you've got arginine, which is polar, and K, which is lysine. And that is indeed polar. Okie dokie. So, non-polar hydrophobic hates water. Hydrophilic loves water. Okie doke. So, even if we just take a look, let's just take a look at hydrophilic and hydrophobic. And by the way, those that are basic and acidic, they are mostly polar. So we're going to treat them as polar. For the purposes of what we're looking at today, we are going to treat them as polar. So if they're basic and acidic, we're going to treat them as though they're polar, which is why they're up here. Hokey doke. So if they're hydrophilic, they love the water. They love being, and inside the cell, inside the membrane of the cell, is a very aqueous environment. So they love being on the outside of the protein. So they're the ones that are hydrophilic, which are your non-polar ones, your polar ones, they will love to be on the outside of the cell. So we can say that these guys are going to be on the outside, these guys are also going to be on the outside, and these guys are also going to be on the outside. But these guys, the non-polar, they want to be on the inside. Now, our amino acid chain wouldn't work. As a folded up protein, it wouldn't work. We haven't got enough polar and non-polars. But just for the purposes of an example, so these guys would be sat on the outside, poking up on the outside, these guys on the outside, and these guys on the inside, because they like to be buried and hidden by the hydrophilic ones who love being out in the water. So I'll just do a quick example. So the cell is an aqueous environment. Hydrophobic amino acids go to the centre of the protein out of the water, and hydrophilic go to the outside of the cell, not the outside of the cell. What an error. The outside of the protein. So essentially, if I just draw a quick, so this is our cell, H2O, this is our protein, H2O, so protein. So I stop calling it a blooming cell. Oop, there we go. So here we go. So these would be where you'll find your hydrophilic. Hydrophilic. They love it. And these would be your hydrophobes. Ooh, wrong one. And these would be your hydrophobes. Okie doke. Now we see examples of these kind of characteristics all the time in the phospholipid membrane. So I don't know if you've come across the phospholipid membrane, but that's obviously, you've got kind of, I'll draw it so it makes more sense. Now these aren't proteins, these are carbohydrates. No, they're not, they're lipids. So here you have water on the outside of the cell. Here's water. And your hydrophobic ones. Ooh. You got your hydrophobes there. And your hydrophilic ones there. Hydrophiles. Okay, so that's just another real life example of the characteristics that can come from being a hydrophilic or a hydrophobic, but they're not proteins, that isn't part of what we're doing. I just thought it would be a real great example. Okie dokie. So now we can move on to bonds found in the tertiary structure. So the tertiary structure, involves the R groups. Now also found, so the bonds that are found in those R groups are your van der Waals, your hydrophobicity and your hydrophilic, which is what we've just gone through, disulfide bridges, which involve cysteine, which is C, hydrogen bonding and more. It becomes a very, very complicated bundle of chemical reactions. It's very, very interesting. Protein folding is the forefront of biology at the moment. We're trying to figure out how exactly they do fold because it's very important. We'll go into that later, why it is so important that they fold properly. So then, for some proteins, some proteins stop here. And sorry, this is an example of a 3D structure of a tertiary protein. So this is actually oxidized myoglobin, which is a cousin, a kind of protein cousin of hemoglobin. So you have your oxygen molecule there. This is your 3D folded protein. So you've got your alpha helix, your beta pleated sheets. And I thought that would just be a great idea. And this is your heme group, which binds to the oxygen. Okie doke. So now we have the quaternary structure. So the quaternary structure is made of different protein subunits that come together to make one structure. So some forces are involved as above. So for example, you have hemoglobin. So you've actually got four. You've got 4 different proteins here, and your heme group. So this is a heme group. And you have 4 different proteins. 1, 2, 3, 4. Okay, so they're the same forces involved in the tertiary structure. So that's when they come together to make one big structure. The proteins that form part of a structure, so many proteins equal subunits. Okay? Alright then, so we're going to move on to how important is protein folding. Now protein folding is very important because structure equals function. Now if the structure goes wrong, it cannot function. And there is no point in having a protein that doesn't function. There's no point in that, it's not going to do its job. So it's incredibly important that it folds correctly. And what happens if it misfolds? Now, misfolding, if it occurs, obviously due to a genetic condition, not a lot essentially, unless of course it causes massive problems, and then you have, the diseases that are caused by misfolded proteins because the instructions aren't correct. But for proteins that misfold that aren't coded for genetically, these are called prions. And I'm sure you've heard of prions. Now they're very dangerous. They are misfolded proteins, and the example of prions, CJD disease, some Alzheimer's. Although it is rare for it to be Alzheimer's, fatal familial insomnia, and things like this. So prions can cause other proteins to misfold. Okay, so they're very dangerous prions. So if it happens, if it's because of genetics, then you'll have a non or not properly functional protein. So an example of this would be cystic fibrosis. Okay, so there we go. So if it's genetic, it causes a non-functional, it doesn't work properly, which unfortunately causes a range of health conditions. And if it happens because it just misfolds, it's got nothing to do with the genetic sequence, it's just a fault, then you have prions, and they're very, very dangerous. Now, I'll just quickly cover what can denature a protein. So you have things like temperature, because temperature is obviously heat, which is energy. And this essentially shakes the protein apart and breaks the bonds. And then this causes unfolding. So this would affect secondary, tertiary, and quaternary structure. Another one is pH, because acid breaks ionic bonds. So this affects the quaternary and the tertiary and quaternary structure. And then we've also got enzymes. Enzymes are capable of breaking apart proteins, and an example of this is pepsin, a protein found in your stomach. So obviously it's part of your digestive system to break apart proteins. So next we're going to cover DNA mutations and their effects upon proteins. I'm going to put that in the same category under the lecture. It's just kind of a little bit extra, and that's going to start preparing us for our practical. So I'll see you there.